a good protocol would, I think, is, six hours of eating and 18 hours of fasting. So for me, today is a day I'm intermittent fasting. I will start eating maybe at about 3 p.m. and I will finish eating. I'll have a nighttime snack at 9 p.m.
So, Dom, how did you end up working with Navy SEALs?
I came in from sort of the back end point, like the Office of Navy Research. They fund projects under a particular program called the Undersea Medicine Program, and that's for warfighter performance and safety. And I was an avid diver as a PhD student, and my postdoctoral fellowship research was really developing technologies funded by the Department of Defense and the Navy that would allow us to study the problems that the Navy SEAL divers have, and also deep sea divers. So that could be oxygen toxicity as it pertains to the pulmonary system, oxygen toxicity as it pertains to the brain, which causes seizures, decompression sickness, nitrogen narcosis, high pressure nervous syndrome. So these are all things that I studied sort of intensely in my postdoctoral fellowship years. And my project was funded to really study fundamentally oxygen toxicity seizures. And we don't know why they occur. And at the time, we didn't know how to prevent them or how to predict them and prevent them. So that led me down a path to basically look into anti-seizure strategies and to, being funded by the Navy is a little different than funded by the National Institutes of Health, where, you're required to just publish high impact peer reviewed papers and get papers out. The military wants a deliverable. So they want some little widget or deliverable at the end of your study to say, look, the guys can take this out to the field and it can apply to them, or they can move it, whatever you develop, into a large animal model, like they study pigs, for example. So, so my research initially really with the Navy SEALs and Office of Navy Research was to do very fundamental science in their basic science program and to develop an anti-seizure neuroprotective strategy. So I was mostly focused on drugs at the time, but I realized that the anti-seizure drugs are really not very good for people with epilepsy or other seizure disorders. And in the process of actually helping a friend in the UK in 2008 or 6, 7, and 8, I suggested the ketogenic diet just by looking to see what do people do who have drug refractory or drug resistant epilepsy. And I discovered the ketogenic diet, which I thought I knew about Atkins, but I didn't actually know the history of the ketogenic diet as a very powerful metabolic based program or nutrition therapy that can prevent seizures. And it was developed in the 1920s. And I decided to pitch this to the military as a neuroprotective strategy. And it took about 2 years from initial thought and studying and pitching it. It took about 2.5 years, 2 years to get funding for that. So right now, going back about 10 years ago, 8 years ago, the initial funding was to understand why oxygen toxicity occurs, uh, biomedical countermeasures that can prevent oxygen toxicity, and then the last 5 years developing specific things, formulas that the warfighter can take by mouth that will instantly produce uh, therapeutic ketosis to enhance their protection against oxygen toxicity, cognitive performance, physical performance, anti-inflammatory effects, because that's a problem too. So we're looking into a wide range of things on a particular, class of compounds and how that can be used specifically in the context of extreme environments. So as it pertains to the undersea environment, but now we're working with NASA in the space environment, Air Force, AFRL, altitude. So that's kind of like the very, very general picture. We have lots of research projects that are run by undergrads, medical students, PhD students, postdoctoral fellows, and then we collaborate with people all over the United States and all over the world, actually. And we have a human study going on at Duke University right now, looking at, for example, nutritional ketosis in humans to prevent oxygen toxicity. I do a lot of rat studies in the lab, but we have actually got to the point now where we're developing and moving some of these strategies into more like human factors training or experimentation.
Wow, I want to talk more specifically about what it is that how this came about that Navy SEALs getting seizures. I don't think people necessarily make that connection with diving. Could you explain these guys having seizures? How does that work? What could be going on there?
Yeah, absolutely. So if you just do a search on central nervous system oxygen toxicity or CNS oxygen toxicity, we know that oxygen toxicity of the CNS is a limitation of hyperbaric oxygen therapy, which has about 14 different FDA approved applications. So you have things like decompression sickness, right? If you get the bends, you got to put somebody in a chamber, press them, and then bring them back up, to surface to prevent little bubbles from forming. That's called the bends or decompression sickness. That's one application. Wound healing is probably the most popular, most used application. Ischemic wounds, diabetic wounds, radiation necrosis as it pertains to cancer treatment, carbon monoxide poisoning. So if you can, for example, think of carbon monoxide poisoning. So the carbon monoxide attaches to the hemoglobin molecule and it's starving your body of oxygen, right? So how do you get that molecule off? It binds very tightly. You got to put the person inside a hyperbaric chamber, press them with the maximum partial pressure of oxygen that you can use, right? But a limitation of how high you can get the oxygen level would be oxygen toxicity seizures. So you can really only go up to about 3 atmospheres of oxygen. So we're right now, I'm breathing air, which is 0.20 ATA of oxygen. It's 20% oxygen. When you're breathing 100% oxygen, that's actually 5 times higher the level of oxygen that you're breathing. And it's not toxic unless you're in a hyperbaric environment. So the partial pressure of oxygen is a function of the barometric pressure and the concentration, right? So you factor those together, and that's hyperbaric oxygen therapy. And oxygen's a drug. It's a very powerful drug, and it at a certain level it becomes toxic to humans. So Navy SEAL divers are kind of unique in that they use a closed circuit rebreather, and meaning that the advantage is a stealth component. They can be underwater and there's no bubbles coming up, right? The disadvantage is that the oxygen concentration is very high. So it's like for a Draeger rebreather and similar units, it's 100% oxygen. So if they go down to just 50 feet of seawater, they have the potential to have an oxygen seizure within 10 to 12 minutes. So that's not a lot of time, right? At just 50 feet of seawater. Typically, they dive pretty shallow, but in the event that the water is very clear and you have people looking for you overhead, or you're taking fire, or you got to dive down to a ship or a bridge and plant a mine, you got to go deeper than that occasionally. So to enhance the safety and performance and success of the mission, we want to be able to ensure that these units can be used without the potential of oxygen toxicity seizures, these closed circuit rebreathers. And we don't know how really to predict it. So if you could wear a little widget that could, for example, detect heart rate variability or EEG activity or respiratory rhythm generation, or we know that there are perturbations in respiratory control that precede a seizure, from animal data. So we're working on little strategies to predict the seizures, but most importantly, we're working on strategies that can give more bottom time to the guys out in the field using this type of equipment and making it safer for them. And the development of these technologies, these countermeasures against oxygen toxicity have utility for hyperbaric oxygen therapy, making it safer and making it possible to allow people to get a much higher concentration of oxygen to treat various maladies or toxic exposures, for example, like carbon monoxide, and to be able to do that in a much more safer way. Or if someone's prone to seizures, perhaps they have a brain tumor, and we've explored the use of hyperbaric oxygen therapy for cancer. But if you have a brain tumor, you may be already susceptible to having a seizure, but being in a state of therapeutic ketosis may give you much more leverage and decrease your potential for having a seizure. So you could potentially utilize a therapy that would otherwise be, counterindicated or contraindicated for that. So we work in a lot of different areas now. My main thing was studying high pressure oxygen, the negative effects of it, and preventing that. And now we actually have studies where we're looking at hyperbaric oxygen therapy for different applications. Right now, they are more or less, off-label use, things like hyperbaric oxygen for cancer. But unless you've had radiation therapy, so it is FDA approved for radiation necrosis. But we think that high pressure oxygen can hyperoxygenate tumor tissue, reverse tumor hypoxia, and shut off some of the gene drivers like HIF1 alpha, VEGF, and various factors that contribute to cancer growth and proliferation. Also, by hyperoxygenating the tumor tissue, it increases reactive oxygen species to the point where you can overwhelm the cancer's antioxidant capacity, and it sensitizes that cancer tissue to other forms of therapy, right? And radiation kind of works the same way. Radiation kills cancer cells through reactive oxygen species. Hyperbaric oxygen therapy just does it more gentle, in a more gentle fashion. So we're exploring the use of, so out of my military funded research came the observation, because we have hyperbaric atomic force microscopy, hyperbaric laser scanning confocal microscopy. We have unique technologies that no one else really has in the world. It allowed us to look at cancer cells, for example, and we do human dermal fibroblasts and different types of cells. We observe that in cancer cells, they overproduce oxygen-free radicals under graded levels of oxygen. And that oxygen would kill those cancer cells, whereas normal healthy cells were resilient against a similar level of oxygen. So that led us down a path which was kind of getting off track in some ways, but led us down a whole path of exploring hyperbaric oxygen and other modalities for cancer treatment as an adjuvant to different therapies we're exploring right now. So a little bit off topic, but that now I can say we have about a third to maybe half of the lab is studying cancer and different projects related to cancer and using hyperbaric oxygen in combination with other things. So that could be a whole nother podcast, but but that project evolved out of research and an observation from equipment that was funded by the Department of Defense with what's called a DURIP grant. It allows us to buy equipment and develop and use that equipment for very novel, somewhat esoteric projects, looking at, CNS oxygen toxicity or the mitochondria of neurons during a PO2 equivalent of a Navy SEAL dive operation sort of scenario.
Wow, Dom, you're blowing my mind. You're all over so many amazing things.
I didn't mean to go off in different areas, but I want maybe your people to know that military research, just like NASA research, has very broad applications, and a lot of the technologies that we use today, GPS, other things, really evolved out of military-funded research.
Absolutely, yeah. My graduate work before I was in the Navy was a study of people with a disease called spinal muscular atrophy, and it was Department of Defense funded. And people don't always make those connections, but it's amazing how it does play into so many different fields of research as well.
Yeah.
And while I was there, I also my principal investigator was Dr. Daryl DeVivo. He studied DeVivo's disease or GLUT1 deficiency syndrome.
Yeah, absolutely. I know Dr. DeVivo.
Oh, you do?
Yeah.
Great
Yeah.
guy.
Yeah, we're actually funded by the Glucose Transporter Type 1 Deficiency Syndrome Foundation. So we have GLUT1D mice where we just published a paper last like 2 weeks ago looking at the effects of anesthesia on the GLUT1 mice. So I know Dr. Klepper, who's sort of maybe his protege, and yeah, I've met with Dr. DeVivo because I present at the GLUT1 Deficiency Foundation conferences.
Oh,
And yeah,
oh
very
wow.
cool
Yeah.
that you know him. Small world.
Yeah, maybe we've crossed paths before then.
Yeah.
But in working with him and on GLUT1 deficiency syndrome, there was talk of the ketogenic diet, and at the time,
Yes.
it was new to me. I just saw these amazing slides where you'd see a child who was having seizures that were debilitating, and then all of a sudden, they'd say then the child did the ketogenic diet, and you see the child walking upright and healthy and alert
Hmm.
and things. Can you explain how there's been a history of this ketogenic diet, that it's not just a new trend in 2018, that it's something that's been around for a while?
Yeah, yeah, it's been around. Well, I mean, fasting ketosis, we could say, has been used for millennia, dating back to the times of Hippocrates, like, 400 BC or more. And it was observed that fasting could, quote unquote, cure seizures. And actually would work for some time after, even after initiating food again. But fasting in that context was really not sustainable, right? Because you have to eat again, and then it takes you out of fasting ketosis. So a lot of the benefits were derived from the metabolic physiological state of fasting, which is a suppression of the hormone insulin and also a decrease to some extent of glucose levels and also an elevation of ketone bodies. And a metabolic based therapy was developed at the Mayo Clinics in the early 1920s. And this was the ketogenic diet, which incorporated a macronutrient ratio that was very high in fat, about 90% fat. And it had adequate amount of protein to prevent protein malnutrition with the least amount of carbohydrates possible, 0 to maybe 5%, if that. And consuming this diet produced uh, metabolic physiology that was in many ways mimicked the state of fasting in a eucaloric diet, meaning that you were getting the calories that you needed to sustain, growth and repair and your weight maintenance. And that this nutritional ketosis was a means to induce therapeutic ketosis, and then that had in and of itself, just through a dietary means, an ability to metabolically manage seizure disorders, independent of the etiology. So, which is interesting because whether you have temporal lobe epilepsy, glucose transporter deficiency syndrome, now we know Angelman syndrome, or absent seizures, even different types of seizures, the ketogenic diet tends to work. And now, even a century later, it tends to work better than the drugs in most cases. When drugs fail, people are put on a ketogenic diet, especially for kids. It's about 2/3 of the kids respond favorably, even after multiple drugs fail. So the question could be like, why wouldn't kids use this first? Because many of the anti-seizure drugs do cause developmental delays that may not be reversible in kids. So if you load a child up with phenobarbital or different types of anti-seizure drugs, it can actually impact their development, their brain development, and then you don't get that back. It can impact their IQ and things. With the ketogenic diet, that doesn't happen. I mean, it may actually have some cognitive enhancing properties, maybe for different subsets of people. So the diet has been around for a long time. Its use was marginalized in the late 1950s, 60s, and 70s, and even 80s when drugs came online. And then Jim Abrams of the Charlie Foundation, the Hollywood producer, his son Charlie was stricken with seizures, was not controlled by multiple drugs, and he was really upset that his son was not offered this as a therapy. And they initiated the diet at Johns Hopkins, and he became very vocal about the ketogenic diet for epilepsy. He was on Dateline NBC in the mid to late 90s, and then Meryl Streep came on and did a movie about the ketogenic diet. Most people don't know Meryl Streep did a movie about the ketogenic diet, and that was called First Do No Harm, and that brought more publicity to the ketogenic diet. for its use in neurology. And it still remains the standard of care in neurology for drug-resistant epilepsy. It's not the first line of treatment, but it works for glucose transporter deficiency syndrome, where there's essentially hypoglycemia in the cerebrospinal fluid because you can't transport glucose across the blood-brain barrier. An elevation of ketones in the blood will readily cross the blood-brain barrier and restore brain energy metabolism and even neurotransmitter production, like in kids and adults that have GLUT1 deficiency. So it is really a life-saving therapy for that disorder. And many other, even metabolic disorders, PDH deficiency. I mean, there's all different types of disorders it's used for. And now we explore its application for a lot of other things. We're looking at Kabuki syndrome right now, and we're starting a new project. Kabuki syndrome is essentially a balance between gene repression and gene expression, and an elevation of beta hydroxybutyrate functions as something called a histone deacetylase inhibitor, which can actually turn on the expression of genes that are otherwise silenced with this genetic disorder. So even in the presence of a persistent molecular pathology, the ketogenic diet, by virtue of elevating beta hydroxybutyrate, which is a class 1, class 2 HDAC inhibitor, can epigenetically enhance and restore normal brain function. In the Kabuki mouse model, it actually it saves the dentate gyrus neurons and rescues them from being degraded. So we are doing work on that now in the lab and just starting that up. And the idea is to rapidly move that into human therapy with kids and elevating that endogenous metabolite that we know functions as an HDAC inhibitor. There's an experimental drug called AR42, but it's not available yet for kids that have this. But beta hydroxybutyrate works just as good or better than AR42, which is not yet available. So our lab is actually working to logistically right now set up everything that's necessary to do a preclinical animal model study that will hopefully parlay that into a human clinical trial on something like Kabuki syndrome. So, basically using ketones as an epigenetic regulator to basically alter gene function to restore some sort of normalcy in people who have this genetic disorder.
And for those who aren't familiar with epigenetics, could you kind of explain? Because I think when people hear ketogenic diet, or even if they know what BHB is, the idea of it functioning as a way that affects genetics is maybe new for a lot of folks.
Yeah. Yep. So think about your your genome, your normal genome. We go to 23andMe and we get our genome, right? And that's kind of written in stone, I guess you could say. That's kind of our genome. Or we used to think that, right? But there are processes that happen in the body that allow us to go in there with more or less a repair kit and alter the genes in a way, or the expression of various genes, to alter those expressions to influence our genome. And we call that epigenetics. So epigenetic is sort of above the genome, right? And it's the process in which that our body has the ability to alter the expression of various genes, and even to some extent change our genome over time. And that could be environmental factors, that could be drugs do this, that could be metabolism, fasting. Now we're realizing that metabolites just are not energy substrates, that these metabolites, like beta hydroxybutyrate in particular, have signaling functions in our body that in some ways can impact the expression or repression of different genes. And we are just going down in that direction. When I started this research 10 years ago, we only knew of ketones as an alternative energy fuel, mostly for the brain. But over the years, we've discovered that they can influence gene expression, that there's a ketone receptor, that they can influence an inflammatory pathway that the drug companies are scrambling to design a drug to inhibit. That beta hydroxybutyrate can do that. So these are all things that sort of have popped out of the data and published, not all in our lab. Actually, most of it is published by other very big, high impact, journals and labs that have really teased out the molecular pathways that have really been elucidated, and more needs to be sort of determined exactly how some of these pathways are influenced. But one of the most exciting one to us is being able to alter our genes, right, through altering epigenetics, which is being able to edit in some ways or influence the expression of various gene pathways.
Yeah. And so for people who want all these benefits of this diet, what are other benefits? I've heard you talk about things with austere environments, your own work with NASA, performance for athletes. How does this change in your diet translate to so many of these things, and what are those things that people could look for?
Sure. Like this research is right now at its infancy. So the use of the ketogenic diet as a means to enhance resilience in an extreme environment. So, I've done enough experiments on rodent models to know that being in a state of therapeutic ketosis makes the rat what we call, from our drug studies, a super rat. So basically, when you put a rat into a state of ketosis and elevate its ketones to the same level that would be achieved after me fasting 7 days, that rat is 600% more resilient against oxygen toxicity seizures. And we've never been able to achieve that with any drug, even like anti-seizure compounds that are so powerful that are not on the market yet because they have, side effects. We can't even get that sort of neuroprotection from that. So this is work done, about 8 years ago. So the question is, how do we leverage this technology? How do we develop it into a way that's palatable, that's tolerable, that is FDA approved? So we're working on that. Well, we're working on the use of these compounds for different childhood disorders, and that may set a precedent or a pathway for us to, get some of the more powerful compounds for use in special operations community, things like that. So we do know that the ketogenic diet has a macronutrient ratio that's mostly fat, and ketones are the fourth macronutrient, I like to say, because they are calorie-containing substances. When we consume them, if we ate nothing else, and studies on dogs have been done where you consume something like 1,3-butanediol, and it can sustain performance on a treadmill for a long time. And that compound ketones are not classified as a macronutrient yet. So there are carbohydrates, proteins, and fat, right? So I think of them as a water-soluble fat molecule that can readily cross the blood-brain barrier. So the advantages to the warfighter or NASA, personnel would be the energy density is quite higher, right? So the ketogenic diet, if we're just looking at macronutrient profile, and I've had some of the engineers do calculations on a 4 to 6 man crew to Mars and back, it ends up saving about 2 large SUVs full of weight. So in food, if it's a 1.5 or 2 year mission. So that's a significant amount of weight because the energy density, you have 9 calories per gram, as you know, for fat, and 4 calories per gram for carbohydrates and protein. So just shifting the macronutrient ratio, instead of 70% fat or carbohydrates, you make that 70% fat. Like right there, you are more than doubling sort of the energy density, right? So major logistical advantages, which the engineers love. I'm interested in more of it as a biological countermeasure against those environments, which could be the undersea environment, as you know, hypoxia or hypobaric hypoxia at altitude. So there's a satiety factor too. So when you're adapted, when your body is fat adapted and what we call keto adapted, the you don't get hungry. So you can, in the absence of food, or if you're in austere conditions and your blood glucose levels drop, you are going to have brain fog and a pretty dramatic, predictable decrease in cognitive and physical performance. If you are adapted to ketones, the ketone levels, when they're elevated in your blood and you go hypoglycemic, you're asymptomatic for hypoglycemia. So you can, even in subjects that were fasted for 40 days and they injected them with insulin, they could readily do cognitive function tests and be fine. So that in and of itself, I think is a major advantage. Obviously, a major advantage is, and there's no other fuel that I know of that could really do that. That could make you asymptomatic for hypoglycemia when there's an absence of food availability. Our bodies kind of do that naturally when we fast, but being keto adapted allows you to do that very quickly. And once you've done the ketogenic diet and go back to a high carbohydrate diet and then go back to a ketogenic diet, you make ketones about twice as fast, because you've upregulated a lot of the enzymatic pathways, ketolytic enzymes, and transporters that readily, it's almost like you activate a genetic program that we call it metabolic memory. When you work out in the gym and you work up to, a 500-pound deadlift, it might take you 10 years to do that, right? But then you stop for 6 months and you get back in the gym again. And about 6 or 8 weeks, you could get back up to that. So your metabolism, I think, and this is stuff we're looking at now, is kind of the same way. Once you've acclimated your body to be keto adapted, that you can readily go in and out. We're starting to see that. It has not been studied. So what I'm saying here, I guess I'll have to stay speculation. But as a scientist, I've done it on myself, and we have some rat data. Hopefully we'll publish that soon with our colleagues. So the satiety factor, ketones are anti-catabolic. That's their evolutionary function. If we did not go into a state of ketosis, we would catabolize all our gluconeogenic amino acids in our skeletal muscle, and we'd waste away in about 2 weeks and die probably of cardiac failure. But because we have essentially an endless supply of body fat, even a lean person has like 20 to 30,000 calories of body fat. That body fat's liberated, converted to ketones, and then those ketones are profoundly anti-catabolic. So they prevent many of the proteolytic pathways, and that they are very protein sparing, we say. So that's their function. So the satiety factor, the energy density, the protein sparing, and some of the stuff that we're looking at now with NASA Extreme Environment Mission Operations. I was on a crew member for 22, which is working with astronauts in an undersea environment where we sort of are looking at a variety of different things. And my wife will be on Nemo 23, and that's coming up. We're preparing for that. So I've noticed in myself that the requirement for sleep is less, about slightly less, maybe about 30 minutes less. And the percentage of deep sleep is higher. So instead of getting, 60 minutes of deep sleep each night or 70 minutes, if I'm out of ketosis, if I'm in ketosis, I routinely get about 90 minutes of deep sleep to 2 hours of deep sleep, 220 minutes. My sleep is very deep, and my sleep architecture, my sleep timing is less, and my sleep architecture is more. And then I've seen a lot of sleep data from people out there suggesting something's going on. So it needs to be kind of studied, and also stress. So, and we've already have several publications on this, and another one that we recently submitted showing using an elevated plus maze, which is used by pharmaceutical companies to evaluate anti-stress or evaluate the fear response in rodent models. We noticed early on that it was easier to handle rats and mice that were in a state of ketosis. They didn't have this fear response. They weren't trying to bite you. So if you use an elevated plus maze where a rat kind of goes out on a catwalk, and they don't like heights, they can stay out in the open arm, or they can go into the closed arm where it's safe. And rats have a natural tendency to explore. And humans have a natural tendency to explore. So when they're in a state of nutritional ketosis, they stay in the open arm, I think about 25 to 30% more. And they explore their environment, the novel environment, so that's the implication there. For humans, it's for people who have like PTSD, maybe like social anxiety and things like that. We think, especially for PTSD, we think that being in a state of nutritional ketosis helps balance the neuropharmacology of the brain. It does this by elevating GABA to glutamate ratio, by activating glutamic acid decarboxylase, the GAD65 and 67 enzyme. So you don't have to know all that, but it changes the neuropharmacology of the brain to make you more relaxed. It's actually hitting things and receptors that actually maybe hit, in a more abrupt fashion with alcohol, but it's doing it, naturally. It's, more GABAergic activity is a more, brain stabilizing, calming activity. And we're also looking at the adenosine receptor and serotonergic systems. But the thing that jumps out and why it may have an anti-seizure effect is that you are taking a potentially hyperexcitable neurotransmitter that's actually could be neurotoxic in the context of traumatic brain injury, PTSD, and seizures. That would be glutamate. So glutamate is converted to GABA through an enzyme called glutamic acid decarboxylase. Ketosis activates that enzyme, so you convert more of this stress, anxiety, hyperactivity neurotransmitter to a more brain stabilizing, calming neurotransmitter. So that's firmly worked out, and we're working out the details as to what receptors are overlap with serotonergic, adenosinergic, and all these other things. So we're working out that pharmacology. So from a warfighter perspective, from an astronaut perspective, the energy density, takes $20,000 per pound to get something up in space. Satiety factor, anti-catabolic factor, sleep and stress, and we're also now. We can't really talk about it, but we are doing studies now looking at physical performance and cognitive performance. From my perspective, I'm interested in physical and especially cognitive performance in the context of extreme environments. So you can do this task at 1 atmosphere of pressure, right? Some, reaction time or critical thinking task, and there's different tests that we use for that. Can you go to, 18,000 feet and do the same task or 20,000 feet or whatever, hypoxic? There's a very predictable, impairment that will be, conferred upon the person or, following this cognitive function task. Can we preserve that under extreme environments, in the undersea environment, as it pertains to nitrogen narcosis, high pressure nervous system activity, things like that. So that's kind of what we're doing now in rodent models. I mean, we will look at the activity under ambient conditions, but we also put them into these extreme environments and see if we can preserve, make them more resilient in that environment. And we're vetting out various types of strategies to induce nutritional ketosis and finding out what can rapidly go to the field. So as far as, tolerability, safety is probably the big one. And, is it something I can put in a container or put in a little 5-hour energy size thing and have a couple hundred calories where I can consume that, and that will give my body a superior alternative form of energy that has a host of other things. And it's likely not going to be a single agent, but it's likely going to be a formula. So now we're working with different formulas. It's encouraging that a single agent can have a profound effect, but we think that the real, the greater effect can be when you start mixing these things together and adding some cofactors. But we're very encouraged by just a single agent has outperformed all the drugs that we've, that have ever been tried for anti-seizure. So we're probably going to take that agent and combine it with various other things to make it more tolerable, palatable, and more efficacious.
Wow. Yeah, the possibilities of the ketogenic diet in a pill or a shot or something like that are huge. I was skeptical.
I was pretty skeptical in the beginning, but the military actually wanted something that can be consumed just prior to mission. And and they're looking at, they didn't want to do the high fat diet. Now our views have changed to the high fat diet over the last 10 years, so that could be revisited. But they really wanted to, it wasn't my sort of decision on this. They really wanted they wanted to induce therapeutic level ketosis independent of dietary restriction. That was what was told to me. And I was like, oh, how am I going to do this? So I saw that NIH was working, that Oxford was working. So I visited, those places and met with scientists. One of them, Dr. Richard Veach, he was the graduate student of Hans Krebs. So one of his best students, he's at the NIH and was actually funded by DARPA with a lot of money to develop a ketone ester for warfighter performance. So we, tested that compound, and instead of just focusing on one compound, which most labs do, we study everything and find out what works, and we throw away what doesn't work, and then we focus on developing novel compounds, testing what's already out there, but also developing new compounds that are more powerful, more dense, and more have greater ability to be consumed and tolerated. So we're at that now. Some of the things are highly palatable and tolerable, but they tend to be the sort of, less potent agents. So the potency of a compound is almost inversely proportional to its tolerability. So as these ketone esters, as you have like a monoester, diester, or a triester, and in different compounds, they tend to get increasingly more aversive in taste and tolerability. So we are working on, that's why formulating is very, very important to us. And we're doing that in rats now. And also I test things on myself too sometimes. So
yeah, the N of 1 experiments, you're famous.
Yeah.
Definitely. So thinking from the perspective of your day-to-day guy in the Navy who sees all these potential benefits, who sees the idea of, I'm going out on the ship and I'd love to be able to, if I have to go a while without food to maintain performance, to get that deep sleep, even if I'm not going to get a lot of sleep, all these things that you're talking about. And now a lot of the folks that I work with, they fly in a particular aircraft that just got aerial refueling capabilities. So now their missions go from 4 hours to 8 hours in a plane with, no coffee maker, no microwave, nothing like that.
Yeah.
How can a normal person take this kind of stuff that we have right now, the technology we have right now, into a shipboard environment and get these benefits?
So I'll approach it first from the perspective of diet and from time-restricted eating protocol, right? So intermittent fasting, that's a big thing, right? I like to call it time-restricted eating, right? So I talked about when you fast or do the ketogenic diet and then you go off the diet and then go back into fasting again, you adapt much quicker to fasting. You could preserve your cognitive and physical performance, because your body starts making ketones faster. So I would suggest that guys do not the ketogenic diet, but do intermittent fasting. A good protocol I think is, 6 hours of eating and 18 hours of fasting. So for me, today is a day I'm intermittent fasting. I will start eating maybe at about 3 p.m. and I will finish eating. I'll have a nighttime snack at 9 p.m., right? It will be my last meal. And I basically just skip breakfast and I'll skip lunch. Sometimes I start at like 1 p.m. and finish at like 7 p.m. And that's 6 hours of eating. And I'll typically eat maybe 2 meals and maybe a snack. And then that 18 hours of fasting does put us in a mild state of ketosis, even if we're not like, on a ketogenic diet. But if you do low carb or if you eat ketogenic during that eating window, then you're kind of always getting some of the benefits of ketosis. When you start eating again, you still maintain, a mild state of ketosis, and then you get sort of the benefits of fasting. So that coaxes your body into being what we call fat and keto adapted. So if you take that individual and you take away and you have limited food availability in an austere environment, that individual will have better access to his or her brain will be more resilient to hypoglycemia and also have a greater capacity to liberate fatty acids from adipose to preserve their energy, cognitive, physical performance under high energy demands. So that's sort of the advantage right there. So that's like low tech, right? That's like you know your macronutrient ratios and time restricted eating, and then you have exogenous ketone supplementation, right? So this could be something that's in like a little 5-hour energy kind of thing, and it's a very energy dense formula, perhaps of ketogenic fats and exogenous ketone, mineral ketones, which could be beta hydroxybutyrate, attached to sodium, potassium, calcium, magnesium, which is a salt, so it gives your body minerals. And then there's different esters that could be utilized, and there's a whole toolbox of esters that we're studying right now. The important thing is that you could drop guys off for 5 or 6 days into an area and let them do their mission, and they could carry on their bodies various small vials of this stuff and be able to consume it, and it could be formulated in a way to give them not only a burst of energy but sustained energy over a fairly significant period of time. If they were to get a blast injury, traumatic brain injury, or if they were going to do an oxygen rebreather dive, if they're going to hypoxia, like they would potentially have neuroprotection under all those different circumstances and may be able to maintain cognitive resilience a little bit better too under those circumstances. So these are things that we are doing in animal models that we want to do in human studies. So the trick with human studies is finding the optimal formula that they can tolerate, the dosing, the individual, and it takes, rats, you have an inbred strain of rat and they're all kind of the same and the data looks great, and they have humans and this guy's eating, basically, monsters and sugary drinks and Snickers bars, and another guy's already doing paleo, as he may respond differently. So we really need to get funding to be able to do this in a very controlled environment, because the payoff could be pretty big as far as warfighter performance. It'll probably start in the special operations community and maybe work its way down. With the idea is that you have this as a tool in the toolbox. I'm not all about replacing what the military is going to eat as far as their nutrition, because that's not going to happen. But to let them know that a ketogen or low carb eating strategy is a strategy that may have these benefits, and to make it available for those guys who want to do it. Knowing what we know now, the rodent data is very compelling, and the stuff that we're doing now is very compelling, and it's already moved to human studies that are ongoing. So I see a lot of potential there as using nutrition as a performance, not only as a performance enhancer, but something that can enhance the safety too. That's the primary objective, the safety. But out of those safety studies also led us down paths where we're looking at performance and performance resilience. So there's different ways to do it. Like I could talk about, different types of formulas for exogenous ketone formulas, but I think we're a little bit too early yet in describing the ingredients in those formulas. We're in the basic science concept of that, but time restricted eating, low carb, ketogenic diet, exogenous ketones, maybe all in combination too, right? So maybe not to do one or the other, but these things likely have either additive or synergistic effects when used together.
Yeah, so many tools in the toolbox, like you said. Mm-hmm. And for people who are in general just trying to see what works for them and what doesn't, you're famous for these N-of-1 experiments where you're testing yourself, you're pushing your limits, and now so many people are wearing monitoring devices, they have access to lots of data. What advice would you have to people who want to experiment and to see what's optimal for them? And maybe could you also tell the story of your 7-day fast and the deadlifting?
Yeah, that gets around. Tim Ferriss mentioned, I didn't know he was going to mention that on the podcast, but I guess Peter Attia told him about the story. So monitoring. So what can the person out there listening right now do? They can go to their local CVS, Walgreens, drugstore, Amazon, and buy a glucose and ketone monitoring system, right? And they can it would be good to know their glucose response to a meal. That's very important from a health perspective. You don't want to have glucose excursions up to like 200 milligrams per nanometer. You want to stay, under 120. So I would say try intermittent fasting, perhaps the time restricted eating protocol with low carbohydrate diet, perhaps starting at 100 grams or less per day and then working down to 50. And over a period of time, you can check your blood ketone levels. And once you're above zero point 0.5, you are clinically in a state of ketosis. And for a type 1 diabetic person who's not on a ketogenic diet, that could be dangerous. But for us, that's a good thing. So diabetic ketoacidosis is very, very different than nutritional ketosis. And my PhD student, Andrew Kutnick, did a TEDx talk that you can look up, which talks about that subject, a type 1 diabetic using nutritional ketosis to manage his disorder. So that's all I'll say about that. But I get a lot of questions about type 1 diabetes. So I just, refer to Andrew's TEDx talk. So for nutritional ketosis, you want to be 0.5 or above, ideally around the 1 to 2 millimolar range. Check your ketones typically later in the day when you've had some ketogenic meals in you or you're fasting, and see how you feel. And also maybe when you're not measuring, see how you feel subjectively when you have the most energy, when you have the most lucidity and mental resilience, I guess you could say. When I get into these phases, I'll do a lot of writing or a lot of sort of tasks that require a lot of cognitive, and I will tend to check my glucose and ketones, and I typically find that my ketones are in like the 1 to 2 and maybe upwards to the 3 range. And some people I know actually get much higher. There's people in the lab that are like running 4 or 5, and other people that are on a strict ketogenic diet and can barely even make it to that 1 millimolar range. So everybody's going to be a little bit different. So buy a meter online. They're relatively cheap, under $50. The strips, if you search around, you can get them for anywhere between $1 to $2 a strip, and get some data on yourself, experimenting yourself. And then from there, you can start evaluating other things. So I use an Oura sleep ring for sleep, and the Oura ring monitors a number of different factors. Most importantly for me, it's a pretty good measurement of your sleep time and sleep architecture, like how much delta sleep you're getting and REM sleep and things. And we use this for our NASA extreme environment mission operations sort of mission too on the crew members. So it's a very hardy technology. I mean, you can train with it, and it'd be hard to break it, actually. So monitoring your sleep with a Fitbit or an Oura ring, measuring your glucose and ketone levels, I think is a good place to start, right? And then we all have certain things. Like I know my strength. I keep a training journal, or used to keep, very detailed records of training journal. And then you could, whether you're a runner or a cyclist or whatever, start monitoring this, get your baseline, initiate the ketogenic diet or exogenous ketones or intermittent fasting, whatever you want to do, and monitor your blood ketone levels, and then assess those things that you're interested in, your performance times, not just subjectively, but objectively, right? And monitor your sleep, because your sleep will be a factor in those, performance too. Get general blood work, CBC, CMP, get a lot of baseline data, because it's going to be important. I think for me, I never thought I would continue with the diet. I was just doing it from an intellectual perspective, in that I wanted to understand what it felt like for my brain to run off a different energy source. So I became obsessed with this about 10 years ago. And I was very inspired by the work of George Cahill at Harvard Medical School, where he fasted subjects for 40 days and was able to, I had some conversations with Dr. Cahill. He passed away in 2012, and a lot of the icons are up in their 80s and 90s, pushing 100 now, and many of them are still alive. They did go on to live extremely productive lives and contributed massively to metabolic physiology. We just don't have the metabolic physiologists nowadays that we had back in the day. But I was really inspired by his work and actually wanted to fast for 7 days, not 40 days. So I did quite a lot of blood work before, during, and after, and was able to get my blood glucose down to a level that was significantly below my ketone levels. So my ketones were at the end stage about double what my blood glucose was. And because ketones can readily cross the blood-brain barrier, we can say that roughly 2/3 of my brain energy was being run off ketones. And during that time, I actually worked on a lot of grants and was very productive and actually got funding off of one of the big grants I worked on during that time. I was teaching, and even at the end of it, I went to the gym and tested my strength and found that like my strength did go down a little bit, but I didn't push myself too hard because I knew, I was thinking I'm fragile. I just wanted to be kind of cautious as to moving weight under that kind of condition. But I did find, which was really interesting to me, that my strength did not take a big hit. And I was semi-keto adapted because for about a half a year to a year or so, I had been tinkering with the ketogenic diet, the clinical ketogenic diet, and it really made fasting pretty easy. The third day I was kind of hurting, but after 7 days, I mean, I was lucid enough to be sharp and give lectures and had the energy to move, 5-6 plates on a deadlift. I never thought going into this, I thought, after 4 or 5 days, you'd be so weak. It'd be hard to stand up, that you'd have, and I did have orthostatic hypotension in the beginning. So I had to I would have soup broth, but no calories. I'd have sodium and water and fluids and things like that to stay hydrated.
For those who don't know, orthostatic hypotension that's when you stand up real quick and you feel dizzy, just for the listener.
Yeah. That happened a lot with me in the beginning.
Yeah, I get that really fast too.
Yeah, your insulin goes down, right? So insulin's role is sodium reabsorption in the kidneys, right? So if your insulin level's down, you're dumping a lot of sodium. So I think in the beginning, I was drinking a lot of water, but not getting the sodium that I needed. And once I started getting the sodium in, it's like I started kind of waking up. My blood pressure came back up again. I remember kind of sodium loading before, I had to do a thinking task or work out or something like that. But I would go for like long walks, at the time, just very easy walks, and that my ketone levels would get up. I'd come back and my glucose would be in a range that the meter wouldn't even detect. It would just say low. It would just say low, and it wouldn't even detect it. And I tinkered with a few things to get my glucose down really low. I don't know exactly how low I got it because the meter wouldn't register it, and and my ketone's pretty high. And realize that this really does make you resilient. Your body feels kind of numb in a way, but at the same time, your senses are heightened. So I would go for a walk and I could smell things or maybe even see things sharper than I could otherwise. So now I got the full understanding of when people fast and they say it brings them to another level. And I think the more you do it, the easier it gets, and probably the more benefits you derive from it too. So, I mean, you could tell major things were happening in your body, major things are happening like autophagy. Of course, when you force your brain to go from glucose to ketones, luckily the brain is incredibly metabolically flexible in its fuel utilization, and it can make that switch. Not everybody can. I mean, some people may have a harder time than others, but like I said, the more you do it, the easier it gets and the more benefits you derive from it. And I think it can be a very, I did it for scientific sort of reasons, but it's also sort of a personal journey too. So you learn a lot about yourself, what you can and can't take, how resilient you really are. And you think, man, if I was dropped off, I got lost or something and had limited food availability, at least I know, I could get through it and not die after, 2 or 3 days of not eating. So that was, I encourage people to do that, but I don't make the recommendation. I'm not a medical doctor, right? So not telling people to go out and do it. So.
Yeah, no medical official advice dispensed here. But
yeah.
The general thing, and you talked about how there are these great scientists who inspire you, who have made a huge contribution, and you are someone who is also very active. You were on this big journey. You were making contributions left and right. What is it that drives you to keep pushing on all these fronts?
I think there's a lot of things, that drive me. My students drive me. I mean, seeing their passion for science and looking back and thinking, remembering how fascinated I was, for example, with the brain. I was fascinated, what is a thought? What is that? So I would, as a neuroscientist, as a PhD, I did patch clamp electrophysiology, which is kind of sort of a technique where you record directly from neurons and how they communicate to one another, and you delve very mechanistically into all that. So asking questions and being very curious, and I think from some extent, there's a sort of a selfish, if you want to call it that. I want to be able to learn and extract information from the body of knowledge that's out there, but also from my own experience that I could apply to myself. And I get so enthusiastic and even giddy that I want other people around me to follow it too, and family members, because you become so aware of the benefits of some of these things, even from especially from a nutritional standpoint, where my emphasis was really on drugs in the beginning, and I was kind of more, and we still do drug research, but this idea that altering your metabolic physiology through changing your macronutrient profile can change fuel utilization in your body, and that can have this host from cognitive to anti-inflammatory effects to potentially tumor suppressing effects to anti-seizure effects, like that you can do that you have you are empowered to be able to do that just by the food that you eat, or the different supplements too that could potentially be used. So I'm pretty motivated by that, and I really feel, not necessarily the case for everybody, but because I don't think some of the pioneers in this area necessarily fasted themselves or experimented on themselves, but a lot of them did. And I feel that you really have to immerse yourself into what you're doing and almost be part of what you're doing to really have that enthusiasm to the point where you're so excited it's hard to fall asleep, or you wake up and you're like, kind of, what's going to happen today? Like, what will this blood work show? What will this measurement, like, how will this day go? So to be so immersed in your research that it becomes sort of part of who you are and part of your envisioned contribution, right? So you want to, and it also guides my research. So a lot of the things that I do on myself, or maybe see others do, I get a lot of feedback from people too. That actually guides my research. Someone will email me about this or that, and that has actually guided, various research projects that we have. So every day I wake up and I'm excited. I don't know what's going to happen. Like, I'm usually testing something new on myself. My students they're in the lab now, in the trenches doing research. I get to come in and they have, so much enthusiasm about what they do. So that's kind of what drives me on a day-to-day basis, and being able to look forward and actually contribute to the scientific body of knowledge to use food as medicine. And it doesn't have to be a macro. It could be like, there's companies out there that are creating what I call engineered food, whether it be a cookie or a brownie, where they're not only changing the macronutrient profile, but they're incorporating different ingredients that make it like a functional food, whether it be a prebiotic fiber or probiotics, or an alternative energy substrate. I mean, one of the first things I used was Cytomax. When I rode my bike, it had alpha L polylactate in it, which was that got me interested in alternative fuels. And this is going back 15 years ago. And creatine monohydrate. I started using that in 1991 or 92, and always stayed up on the literature. So I think of ketones as kind of like the next creatine. But much more versatile. So it can be neuroprotective. It can it enhances your body's ATP production, but it has a lot of advantages and a utility that something creatine doesn't have. But mixing them two together would be sort of one of the formulas that I'm working on now. Like, putting various alternative energy substrates together and cofactors, which allow you to use these things in a more enhanced fashion, I would say. So that's an exciting thing. And also cancer. I mean, a lot of what we do, I didn't talk about it much. What could be more exciting than killing cancer cells? Our soldiers go out to war to kill the enemy, to protect us. And I kind of think of my PhD students as sort of soldiers, warriors that are kind of attacking cancer cells with the therapies that they're developing together as a group and testing in cells, in tissues, in animal models, and we're moving it to human clinical trials. So we're sort of on a war against cancer in a very targeted fashion from a metabolic standpoint.
Yeah, and everyone knows someone affected by cancer, so it's such
Absolutely.
a universal cause, and it's great that we've got you on the team. Um,
And the people, the students, and many of the people in the lab were motivated from a personal perspective, and that's why they chose, their particular projects too. That's often the case.
oh yeah, I can get that.
Yep.
Dom, I want to be respectful of your time. Before I ask my last question, where can people find you online?
The best place to find me is the website Keto Nutrition. Most importantly, it's .org, not .com. So ketonutrition.org. And I have a blog there. I have products that we've tested. I have resources like doctors, consultants, podcast. That's my kind of one-stop shop for people who want more information about what we do.
Great. And Dom, my final question. If you were
Mhm.
talking to a military leader who wants to create a culture where people have this intellectual curiosity that you display, this self-experimentation, this drive, what suggestion would you
Mhm.
give them?
I was talking to a military leader to embrace this culture. Uh, I would ask that military leader and try to persuade them to do it themselves and to evaluate whether they thought personally that this was a viable strategy. So I would do something as simple as time restricted eating. I was locked into a pattern of not only eating like 5 or 6 meals a day, I would eat like 7 or 8 meals a day, wake up in the middle of the night and eat a protein bar or shake and go back because I thought I needed that. So I come from that mentality. But when you liberate yourself from these frequent feedings or free yourself from it, it becomes very liberating, right? When you free yourself from that eating pattern. So when it comes to nutrition, maybe we do a lot of different things, but the most simplest low tech thing I can think of is to say, hey, if you haven't tried it before, maybe try time restricted eating. Don't do it every day. Maybe just do it like twice a week and see how you feel. And the more they do it, the easier it'll get. And then a lot of that great feeling that they have towards the end of the fast, that's actually due to beta hydroxybutyrate, the ketone body that I study. So, and that's conferring many of the health benefits like anti-inflammatory effects and, epigenetic effects and things like that. So that may be a gateway to get them into experimenting with an eating paradigm that, without question, has major logistical practical advantages to guys in the field from a physical, cognitive, and maybe even anti-anxiety perspective. And we know, if you're edgy and you're jacked up on caffeine and edgy, so, I go to the range, I shoot quite often, and I experiment on things. I don't talk about those things on podcasts usually, but not only the gym, but I'll go to the range and I'll evaluate what gets me better scores on my shooting tests, right? And I could tell you that the things that I talked about and things that I mentioned, start doing it and if you go to the range, start recording your scores, and you're going to see that too. And that's, something I never really talked about, but I think from their perspective is a very practical benefit too. So the worst thing to do for a soldier that has a gun and shooting is to be sleep deprived and jacked up on caffeine. So, and I've actually been in that situation where I've evaluated my shooting ability under those conditions, and I was all over the place. But being jacked up on caffeine, it gives you the confidence. You actually think you're shooting well until it gives you like a false confidence. But when you're in a state of fasting ketosis or nutritional ketosis and you've maintained that, I guarantee not only physical cognitive performance, but things are very important. Your decision making and shooting performance will improve. So I would say give it a try, but they kind of, for me, I can read all the studies in the world, but I needed to actually experience it myself to have the belief, and that belief through self-experimentation led to a further enthusiasm to pursue this path as my career. So over the last 10 years, that's what I've been doing.
Dom, thank you so much.